when you picture the protective wrap around your brain, you know, the meninges, you probably imagine this passive shock absorber, right?
Exactly, like biological bubble wrap.
Right, just a layer of bubble wrap keeping your brain safe inside the skull. And I mean, we've thought of it that way for centuries, but what if that bubble wrap was actually like a bustling construction zone?
It's completely changing the paradigm.
It really is. What if it dictates exactly how the physical structure of your brain forms and acts as a fortune teller for your future health decades down the line?
Well, it forces a complete rewrite of how we understand neurodevelopment. I mean, we're moving away from looking at the brain as this isolated supercomputer that just, builds itself, to realizing that the packaging around it is actually the master architect pulling all the strings.
Okay, let's unpack this, because tracing exactly how those strings are pulled is our mission for this deep dive.
Right.
We're looking at a landmark July 2026 paper from Cell. It's called Decoding the Spatiotemporal Development of Human Meninges. And we are exploring every conceivable detail of this incredibly delicate phase of pregnancy from gestation week 6 to 23.
And to figure out what the brain's security system is doing at this stage, the researchers used single-cell RNA sequencing and spatial transcriptomics.
Which is just a wild technology.
It's a phenomenal leap. I mean, spatial transcriptomics, things like MERFISH and CTRSEC, they give us the actual blueprint. Previously, we just ground up tissue and lost all the geography.
Right, like having a list of materials for a house, but no idea where the walls go.
Exactly. Now we can look at a microscopic slice of fetal brain and see, exactly which gene is turned on in which cell, right where it sits in 3D space.
So using that tool, the researchers looked at the 3 distinct layers of the meninges. And before we get into the weeds, I want to give you, the listener, a quick mnemonic to picture this. Think of the acronym PAD-P, AD.
That's a great way to remember it, working from the inside out.
Right, so P is for pia mater, which is closest to the brain. A is for arachnoid in the middle, and D is for dura mater, the tough layer closest to the skull. So PAD.
And the spatial mapping proved that these 3 layers, which are mostly built by fibroblasts, they don't just appear all at once. It's a highly asynchronous process.
Wait, asynchronous? So they don't just form together as the skull grows?
Nope. The blueprint demands they form at different times. The pia mater, that innermost layer, forms earliest. We see it expressing extracellular matrix genes like COL15A1 as early as gestation week 6.
Week 6. That is incredibly early. I mean, the brain is barely a tube at that point.
It is. The outer layers, the arachnoid and dura, they're barely getting started. They don't really catch up and stratify clearly until around, GW 12 to 16.
Okay, so the PLA is the foundation first.
Right. And as they separate, they get specialized. The arachnoid turns on transporter genes for the blood-brain barrier, like SLC22A6. And the dura mater turns on ossification genes, like MGP, to interface with the skull. Eventually, they all express this barrier gene, CLDN5, to seal the border.
But hold on, I want to push back on something from the source data here, because if these fibroblasts are just structural, why did the data show the pia and arachnoid heavily expressing neurotransmitter transporters?
Yeah, that's a big discovery.
I mean, we're talking about things like SLC6A1, which is JAT1 and JT2, plus classic synapse genes like SYT1.
Wait, so the security guards at the door aren't just standing there, they have walkie-talkies tuned into the brain's private staff channel.
That's exactly what they're doing, because they're building a dynamic environment. By expressing those genes, they're listening to the chemical chatter of the developing brain to maintain neural homeostasis.
Wow.
And not just neurotransmitters, they also highly express lipid metabolism genes like APOE and APOD. They're literally managing the fat supply to support barrier development.
Okay, let me do a quick recap and a bullet summary for you listening, because that was a lot of dense biology.
So, bullet 1. The meninges have 3 layers, pia, arachnoid, dura, PAD.
Right.
Bullet 2. Development is asynchronous. The pia forms earliest at GW6.
Exactly.
And bullet 3. These layers don't just protect, they actively listen to brain chemistry and build the barriers. So I guess the question for you to think about is, have you ever considered that the packaging of an organ might be just as chemically complex as the organ itself?
It's wild to think about. And it gets even more complex when we look at the immune cells.
Let's shift to that, because the meninges are absolutely packed with immune cells. The source identifies 10 distinct clusters. And over 50% of these are meningeal macrophage, or MMs.
Right, the frontline defenders. But the researchers broke these MMs down into 4 distinct subclusters.
Yeah, okay, break those down for me.
Sure. First, you have the proliferating MMs. They express MKI67. They're just dividing to expand the workforce.
Makes sense. Head gets bigger, you need more workers.
Exactly. Second are the IFH-MMs. These are inflammatory gene highly expressing macrophages. They express S1000A9. They're the classic responders.
Okay, so the fighters.
Third are the AHMMs, which are angiogenesis highly expressing. They express VGFA, so they help build blood vessels.
Got it. And the fourth?
The fourth is the really big discovery here, the CHMMs. These are collagen gene highly expressing macrophages, marked by COL1A2.
Wait, a macrophage expressing collagen genes, that's structural.
Right. It's a newly discovered meningeal-specific population. The meninges are a dense, fibrous, collagen-heavy environment. So these immune cells have uniquely adapted their gene expression just to operate in that specific microenvironment.
Oh, wow. But wait, how do these meningeal macrophages differ from the microglia inside the brain itself, if they both come from the same early embryonic immune cells?
They behave totally differently. Microglia migrate into the brain tissue and settle down. They're stationary. But the MMs highly express a cytoskeleton protein called vimentin, or VIM.
Vimentin, okay.
Yeah, and that makes them highly mobile. They squeeze through the extracellular matrix without breaking the barrier seals.
Okay, so imagine microglia as the local beat cops assigned to a specific desk inside the brain, while the MMs are this highly mobile SWAT team constantly patrolling the perimeter.
That is a perfect analogy.
Let's do another quick recap. Bullet 1. Meningeal macrophages make up over half the immune cells in the meninges.
Yep.
Bullet 2. Researchers discovered a brand new subtype, the collagen-expressing CHMM.
Right, unique to that area.
And bullet 3. Unlike stationary brain microglia, MMs are a mobile patrol unit. So my question to you listening is, if you were designing a defense system for a biological supercomputer, wouldn't you want a mobile, adaptable firewall just like these MMs?
You absolutely need one.
But that raises a logical question. If these MMs are moving around all the time, how do they know exactly where to go?
Yeah.
The spatial data showed they preferentially localize in the leptomeningeal zone, the LZ.
Right, which encompasses the pia and arachnoid.
And to figure out why they park there, the researchers used cell chat analysis to look at chemical signaling.
And this brings us back to the pia fibroblasts, right?
Exactly. The pia fibroblasts act as directors. They secrete a specific chemokine called CXCL12.
CXCL12, okay.
And it turns out the immune cells, specifically those inflammatory IFHMMs and the blood vessel building AHMMs, they possess this specific receptor for it called CXCR4.
Here's where it gets really interesting. It's like the pia mater is acting like an air traffic control tower, right? It shines a chemical runway light, that's the CXCL12.
Right.
And the immune cell airplanes, which are equipped with these CXCR4 antennas, they just follow it down to land precisely in the leptomeninges.
That's exactly how it works. And they proved it rigorously.
Yeah.
The scientists created genetically modified mice, CXCL12 CKO mice.
Conditional knockout mice, right?
Yes, so they knocked out the CXL12 gene, but only in the fibroblasts.
Wait, so the rest of the mouse could make the chemical normally, they just turned off the runway lights in the meninges.
Exactly.
Yeah.
And the MMs completely failed to migrate to the leptomeninges. They were lost.
Oh.
They even proved it with human cells using transwell assays. They put human pia fibroblasts in a bottom chamber, human macrophages in a top chamber, separated by a porous membrane.
Let me guess, the fibroblasts physically pulled them down.
Literally. The macrophages sense the CXCL12 and squeeze through the microscopic pores just to get to the fibroblasts.
Okay, recap time. Bullet 1. Immune cells gather specifically in the leptomeningeal zone.
Right.
Bullet 2. Pia fibroblasts guide them using that CXCL12-CXCR4 chemical pathway.
Yep. The runway lights.
And bullet 3. Without this chemical beacon, the immune cells get totally lost, which proves the pia's role as a spatial organizer. So think about your own body's internal navigation. How amazing is it that cells blindly follow invisible chemical breadcrumbs to build the architecture of your mind?
It's credible. But getting them into position is just step one.
Right, because my next question is, okay, the pia guides the immune SWAT team into place, but what do these immune cells actually do once they're there?
They don't just sit there. Let's look at those IFHMMs, the inflammatory responders. They sit right next to the brain's marginal zone.
Touching the brain.
Exactly. And these specific macrophages express a membrane receptor called TREM2.
Okay, TREM2. I've heard of that with Alzheimer's.
Usually, yes. But here it's a developmental sugar. When TREM2 is activated, it regulates a transcription factor inside the macrophage called NFYA.
Let's trace this chain reaction. So TREM2 triggers NFYA.
Right, and NFYA physically binds to the promoter region of the CCL2 gene in the macrophage. This causes the macrophage to secrete the CCL2 protein.
And where does that CCL2 protein go?
It goes right into the brain cortex, targeting a very specific type of cell called a Cajal-Retzius cell, or CR cell, via its ACKR1 receptor.
And CR cells are crucial, right? They're like the foreman for the brain.
Absolute critical. CR cells position other neurons. If they don't develop right, the cortex is misaligned.
Yeah.
And the researchers show that if TREM2 is knocked out in the macrophages, the CR cell development in the brain is impaired.
Wait, let me ground this in a real human clinical case mentioned in the paper, because this is heavy science. Let's talk about Nasu-Hakola disease, or NHD.
Yes, NHD.
It's a rare condition caused by a deficiency in that exact TREM2 receptor. So if this pathway is broken from the start, you'd think babies will be born with massive brain defects.
But what's fascinating here is they aren't. NHD patients don't show massive brain defects at birth. They actually suffer from adult-onset neurodegeneration, usually hitting in their 20s or 30s.
Oh, wow. So the TREM2 macrophages don't cause an immediate collapse. They just lay a slightly brittle foundation.
Exactly. They establish long-term structural vulnerabilities.
Right.
And meanwhile, those other macrophages, the AHMMs, are sitting right next to them, secreting VEGF-A to signal endothelial cells to build the brain's blood vessels.
Yeah, they're building the physical scaffolding and the plumbing at the exact same time.
Yes.
Okay, final recap for this section. Bullet 1, TREM2 positive macrophages secrete CCL2 via the NFYA transcription factor. Bullet 2, this regulates Cajal-Retzius cells, which organize the entire brain cortex.
Yep.
Bullet 3, deficiencies in TREM2 lead to Nasu-Hakola disease, proving early meningeal development sets the stage for adult health. And bullet 4, macrophages help build the blood vessels. So the question is, are the seeds of adult neurodegenerative diseases planted in the bubble wrap of our brains before we are even born?
The data strongly suggests they are.
So what does this all mean for you listening? I mean, the meninges aren't just a passive physical shock absorber.
Not at all.
They're the chemical architects, the immune commanders, and really the fortune tellers of our brain's future. The asynchronous layering, the CXCL12 beacons, the TREM2 pathways, they prove that you are a masterpiece of cellular communication.
And I want to leave everyone with one final thought to mull over.
Let's hear it.
If the protective packaging of the brain is secretly pulling the strings of the organ it protects, what about the rest of the body? What other connective tissues or packaging like the pleura around the lungs or the pericardium around the heart are quietly acting as the master architects of our health?
That is a massive question. The spaces between the organs might be pulling all the strings. Keep questioning, keep exploring that microscopic world, and we'll catch you on the next deep dive.